All right, so this will be the start of Unit 4, Cell Transport. Um, this right here, like the other ones, has 3 days of notes, and I have a review on there as well. And so we'll go ahead and get started. Now, whenever we talk about cell transport, one thing that we need to make sure that we go back over is the cell membrane. I know we just had the unit on cells, but it's still a really good idea to, and we just talked about what the cell membrane was, but it'll still be very helpful. Sorry, there we go. For us to just go back over it, and then we're going to talk about passive transport today. So this is what we're going to be doing on day one, going back over the cell membrane and then talking about passive transport. So the cell membrane structure, let's try to make sure that we do remember that it's made up of a phospholipid bilayer. That's these yellow small balls that go across the top and the bottom, kind of looks like ping pong balls. And then they have these lipid tails here. That's the phospholipid bilayer. And then, of course, it has these embedded proteins. You might, in this unit, you might start hearing them known as protein channels, carrier proteins, things of that nature. And that's really the main part that you have to understand. So, of course, we have the lipid bilayer. You'll need to know that the balls would be the phosphorus heads. And then, of course, we have the lipid tails, and that would be the legs that look like legs hanging down or going up in the middle. All right. So cell membrane, let's try to make sure that remember that I could be using terms like plasma membrane or phospholipid bilayer. In this unit, you're also going to hear the term selectively permeable. All that does is that just goes back to the function of it. That's just a two-word way of actually giving you the definition of or the function of what it does. To select means to control, and permeable is talking about that things can move in and out of it, but in a controlled fashion. Now, being the fact that it is that we do have all these ping pong balls that which would be like this phospholipid bilayer, those aren't fixed structures where they stay in the exact same place. It's not a rigid like a cell wall is. So that's why it's described as a fluid mosaic. So a good way of thinking about this is if you had like a big pool of water and you put ping pong balls on the top of it, even though it would be completely, you could cover the entire surface of the water of a bathtub or whatever with ping pong balls, and but you could still move those ping pong balls around, but they would still just be there on the surface of it. So that's kind of an idea of thinking of a fluid mosaic. All right. Remember, the function of the cell membrane is that it controls what's going to move into and out of the cell. And don't forget that structure is a phospholipid bilayer with embedded proteins. So there are actually two types of transport. We have passive transport and active transport. Now, we'll go ahead and let you know you will need to know the difference between these two very well. Now, the reason why I have the passive one circled here is that's the one that we're actually going to focus on today. We're going to focus on the passive transport, and in a couple of days, we'll look back at active. But you will need to know how to differentiate between these two, and this is basically how you would do it. So, the easiest way to think of it, of course, active requires energy, and I think that makes sense through the name. Passive does not require energy, no energy is needed. There are three types of passive. We have simple diffusion, which is just, hey, things spread out, kind of like if 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 you're in a classroom and somebody sprays some perfume, people at the other end of the classroom are eventually going to smell it. We also have another type that's called facilitated diffusion. A lot of students get this one confused and think that it goes with active. It does not. Remember that it still has that word diffusion there, so we're still talking about passive. And lastly, we have osmosis. And we're going to get into each one of those individually. Now, on the other side, we have a sodium potassium pump. You actually have a lot of different types of pumps inside of your body. That pump is those proteins. Um, the one that we'll look at more specifically is the sodium potassium, and we're not even going to get that specific with it whenever we do look at it. And then endocytosis and exocytosis, and we'll get into those as well. So first thing to know about passive transport is what is it? So this is whenever substances move across the cell membrane. It can either go into or out of the cell. That does not matter. But whenever it moves across it, we talk about this concentration gradient. And that means it's going to move from an area of high concentration to an area of low concentration. Now, if we were actually in class and we were going over this, I would get up on the board and I would actually draw kind of this image here that we see here on the side. And I think this is probably one of the best images that accurately depicts passive transport. So if you see here, you got this guy, he's up on top of a hill, he has a rock rolls down the hill or the ball rolls down the hill. He isn't having to constantly push it down the hill. It's going to roll down the hill passively. So that's what it's talking about. And if you think about this, where he's standing at is higher, and then where the ball is going is lower. So that is it moving from high on top of the hill to low at the bottom of the hill without needing energy to do that. Okay? So I think this image really helps out with thinking about that high and low side of it, because that's going to be very important in the distinguishing particles moving from high and low, which you will have to do that. So this right here is an actual an image that we had on a test at some point. I think a benchmark actually. So right here, based on this image, I see that I have cytoplasm of the yeast cell. So if it says cytoplasm, I know that that's talking about inside the cell. And over here, we have this solution that's outside the cell. Now, if we're going to look at these particles, we see that we have 5 square dots on the left side, but we only have 3 of these dye molecules on the right side. Well, based on the definition of passive transport, we say it moves from a high concentration, where we have 5 of them, to a low concentration, where we have 3 of them. And also, if you notice, there's no energy being used here. You're going to see what I mean by this in a couple of days whenever I actually show you one of active transport. Remember, anytime there's energy being used, you're going to be seeing that term ATP there, or energy. Okay? So this right here would be an example of that. And you'll see things like this on your test and quizzes. So going over the different types. So first off, we just have just regular diffusion. Now inside of your body, whenever we talk about simple diffusion, we're actually talking about gas exchange. So basically, whenever you breathe in, oxygen comes in, and oxygen needs to get into your cells, but also carbon dioxide needs to leave. They could do that so freely and so easily, it doesn't even get hindered on the way. Now, whenever you have a hindrance in gas exchange, well, that's whenever basically you could have choking and things like that, or you have some type of blockage. But with regular diffusion, we're talking about gas exchange. Okay. Moving on to facilitated diffusion. So facilitated diffusion, much like diffusion, still passive, but now we're moving a bigger molecule. We're not moving something as small as an oxygen molecule and a carbon dioxide molecule. We're actually going to be moving something really large like glucose, but that means that we need the help of the protein channels. Now, I need to make sure that you understand that whenever we say that it requires the help of the protein channels, those embedded proteins that are in the cell membrane, we're not saying that those cell membranes, that those proteins are having to use energy for this. We're just saying that these are so large that they can't go through the phospholipid, so it has to go through these channels that exist in the proteins. All right. Now, if we notice, there are multiple out here outside the cell. There's a high concentration of glucose, but inside the cell, there's a very low concentration of glucose. So that's why they are passively moving through those protein channels. No energy is being used. And that's actually what that term facilitates means. It means that we're getting help, but we're getting help from those embedded proteins. Make sure that you can distinguish that from being active transport. There's no energy being used here. All right. Osmosis. Now, we're going to have a day specifically where we go over each one of these terms that we see here. For right now, all I want you to know that osmosis, the definition of it, is the movement of water across the membrane. Okay? And we're going to, in next class on day 2, we're actually going to go over it specifically because there are certain types of problems that you will see directly or questions that you will see directly using this information. So we're going to go over that next class in more specific detail. But you do have different types of osmotic pressure. We have a hypertonic solution, an isotonic, and a hypotonic. And we'll get into each one of those next class. So some terms to make sure that you remember here. Cell membrane. Make sure you remember that it's a plasma membrane, a phospholipid bilayer. The reason why I put this on here is that many people last year and the year before that had trouble remembering that. If I sat there and wrote a question and I said plasma membrane, they couldn't connect that it and the cell membrane were the same thing. Same thing with phospholipid bilayer. Same exact thing there. We need to make sure that we know that those are synonymous. Selectively permeable. A lot of people kept forgetting this word last year and the year before that. Selectively permeable just means it's the job of the cell membrane. The cell membrane is selectively permeable. It will only allow certain molecules to go across it. So it selects, it chooses, it controls what moves across it. And the last one is carrier proteins. That's those embedded proteins, those protein channels that we use during facilitated diffusion. So there you go. So here's our review for today on passive transport. So first thing, remember we first talked about the cell membrane. So make sure that you can describe the structure and the function of the cell membrane. So please review over that. You need to make sure that you know the two basic types of transport, passive and active, and how they're different. The main thing of how they're different is passive doesn't use energy, active does need energy. And then make sure you know the 3 types of passive and the 3 types of active. So that's what number 3 is talking about, is what are the 3 types of passive. Whenever I ask you to explain each type, what I'm asking here is on diffusion, know that we're talking about gas exchange. Know that we're talking about oxygen and carbon dioxide moving across the cell membrane. Facilitated diffusion, you need to make sure that you know that we're talking about glucose moving across the cell membrane with the help of those embedded proteins. And osmosis is the movement of water across the cell membrane. The last one here is explain what is meant by the concentration gradient. That's whenever those molecules are moving from that high concentration to that low concentration. That's what's meant there. And of course, no energy is required.